On? Yes. Okay. Well, thank you all for coming, even though it's extremely snowy and it's hard getting around right now. Even I don't want to be outside. Today, we have the pleasure of being visited by Dr. Jenn Fehrenbacher from Oregon State University. Just for those who may not have had the pleasure of meeting her yet or haven't heard her background, I'm just going to give a quick intro to her. She did her undergrad at Northern Illinois University, where she majored in geology. She then took some time away from academia and worked in like an IT job, and you had an internship. I forget where.
Oregon National Lab.
Okay.
I worked there for 2 years.
Cool. And then got back into academia when she got her master's and her PhD at University of Chicago.
Yep.
That's right. Thank you. She then went on to do a postdoc at UC Davis and now is working at Oregon State. She originally started in more of the geology paleoceanography side, but now works in all manner of oceanography, both the modern and the paleo. And today we're going to have this moderate discussion. Well, I'll ask her questions, or you guys can ask her questions, and people on Zoom, if you guys have any questions, feel free to ask it in the chat. And we will have it about your origins, your career, your like basically all those things. And I'm gonna sit down too. So
Excellent.
before I get started, does anyone here have a question they'd like to ask to get started? Or I can ask from the, oh, you do. Yes.
I do. Yes.
Get to the ocean through Lake Michigan and
Well, I think you can through the Hudson. If you were to go that route, you could. Yeah.
yeah, I think you wait, how did you get in the system? I assume that was like what you do grad.
Yeah, so for grad school, I studied fossils, and so those can be obtained from any of the core repositories. So when researchers take samples from the seafloor, they go into a core repository. So Lamont Doherty has one. There's one at the IODP, where are they now, Texas or Florida. They've moved. We have a core repository at Oregon State, and so samples from like all over the seafloor, all over the world, even lakes, terrestrial samples are stored in these repositories. And so I obtained my samples from, I think, the Lamont Doherty core repository, and from my advisor who had some from grad school. And so that was, fossil samples can be studied anywhere. And so that's how I really got started, with studying fossil samples. But I do think that you can really study the ocean anywhere. It just requires getting on a ship and getting there for the most part. So Corvallis is an hour from the coast, and I go to sea, for field work. I go to Catalina, and then we go on ships a couple times a year. So I don't need to be by the sea for the most part. Yeah, so it kind of works. Yeah.
If the audience have any follow-up questions from that before we move on? Oh,
Yeah.
Like that morning.
Yeah.
Surface level, or like about your background?
Yeah.
Have you talked about like the polar variety of the
Forams.
Yeah.
Yeah.
Forams yesterday. Have you had a chance to look into ice cores or anything like that?
So there is a species that lives in the sea ice interface, and so they're not preserved in the ice, but they end up being preserved in like sediment samples. People do take them from sea ice to study them. They're really unique. Some of them are they're kleptoplastic. I really wish this person would publish her paper. They steal chloroplasts from algae and take them and use them, which is like the coolest thing ever. And she hasn't published her paper, and I have no idea why. But they live in the sea ice for part of their year, and they also live out in the ocean. It's just a different type. But those are also found in the sea cores. We just don't have as many cores from the Arctic, mostly because it's really hard to drill in the Arctic. It will become easier as climate changes and we have ice-free conditions. And then drilling in Antarctica is also really hard, and then there are just fewer forams preserved there in those regions. Yeah, I hope that answered your question.
Do you want to keep on the origins, the kind of side of things?
Anything. Ask me anything. It's totally fine.
Well, I have this list that people have added too, so just kind of like, what was the trigger that got you into ecology? Like, I know you did a lot of geology work
Yep.
starting up and paleo stuff, but like, what got you into the ecology side of your current research?
Probably that we need to know where they're living in order to use them really well, right?
Yep.
And so, I mean, people have kind of been doing this for forever. We need to know sort of which ones live at the surface, which ones live in deeper water. But then as we've sort of progressed through time, the instruments that we can get, data from and the questions that we can ask have changed, right?
Mhm.
So we can understand better which ones have symbionts and which ones don't by looking at the active chlorophyll. I don't know, it's just changed, I think so. And then looking at how they're responding to climate change has also sort of been a piece of my work, just the proximity to the study site that's in the paper that I think I shared with you, the Lane paper. That was an archived set of samples. And they didn't think that there were forams in them. Super funny story. When they sample the plankton, they swirl the plankton and kind of mix it, and they take a 5 milliliter aliquot. Well, forams are sand grains, right? They're heavy, so they sink. And so she said, we've been doing this for like 60 years, and there are like no forams in the samples, but you can come look. And I went down there with like I have a hand, my hand lens I've had since like 1995 or something. And I sat down in the floor in the shed and went like this, and they were loaded with forams. And so being able to like tease out where they live becomes important for understanding how climate change is going to affect where they go. So I think that's kind of part of it. It's just wanting to know more about where they live means we need to understand their ecology.
Yeah. Have your research interests kind of evolved, not just like from undergrad to like your current position, but like as you are in your current position, like where have your research interests taken you in your path?
Oh man, all over the place. Yeah, no, really all over the place. I think part of it is I can answer questions with tools, right? And so sometimes I end up doing a research project because I can do the analyses on something. And so it's still mostly forams, but the questions that I might be asking are a little bit different because an analytical tool changes. We've also had really interesting, so I have a sediment trap, and I don't remember if I've told someone about this, but we have a sediment trap off the coast of Oregon. And we sort of have this relationship when we've been at sea between the presence of salps in the water column. Salps are colonial tunicates that they're filter feeders. And basically when we do plankton tows and there are salps in the water, there are literally no forams. And so we just figured that this was sort of anecdotal for a while, and then had the opportunity to do 2 sets of surveys in the month of May on 2 separate cruises. One of them was a long transect off the shore, and the other one was a bunch of transects like a month later. And there was just this massive prolonged salp bloom. And salps are these colonial tunicates that kind of can live everywhere, and they're thought to be something that's going to sort of take over in the ocean as climate changes. And so we found this relationship. My postdoc went out and did a whole bunch of plankton tows. And then I have a sediment trap. So we started looking in the sediment trap for fecal pellets, which are the salp's poop. And so I had undergrad students picking through these sediment trap samples, counting salp poop. Like, this is not anything that I thought that I would be doing. And then taking this salp fecal pellets and then dissolving them and looking for shells. And we found that they have juvenile forams and juvenile pteropods in them. And we have the flux of the salps go up and the flux of the forams crash, and then they don't recover. I don't even know if they've recovered because we haven't finished processing the samples. But so now I wrote a proposal to study the correlation between salps and fecal pellets and forams and sediment traps. And so that's not something that I thought that I would be doing, and then that would be like looking at what's going to happen in the future, because salps are supposed to become more common. And then that will change the calcite budget to the seafloor. So these things end up just being like weirdly, like, this is not something that I thought that I would be doing. And I take people with me. So this is Brittany Hupp is working on this project. So it makes some of the people that I collaborate also do stuff that's a little bit different. So I think it just evolves based on serendipitous findings. And that's sort of, I feel like, has been a significant part of my career. It's just like finding something and trying to figure it out. And so, yeah, I think things just kind of keep evolving. It's pretty fun.
Interesting as a grad student to hear that. Like, I feel like I'm like almost set in my ways when I like choose a path, and then to see you can like branch out into so many different avenues is really
Yeah.
exciting to hear.
I think you should
Yeah.
branch.
Yeah.
Yeah. Because it's, first of all, it's fun and it also diversifies, if you end up in a position where you need to obtain funding, it
Mhm.
diversifies where you can get your funding from.
Very true.
And I've had a very, very hard time diversifying my funding. I get almost all of my funding from the National Science Foundation.
Mhm.
And that's been difficult because they can't just keep funding me in the same program. And so I've had a harder time with that. And recently just got funding from the NOAA.
Mhm.
And so having, some diversity is good in terms of funding, and it also makes things fun.
Any questions from the audience? Before we ask our question? What other ones are? Has any of your research or like work influenced like not just your like academic career, but also your perspective on life, or like any like led to any changes in your life you think of?
Oh, that's a good question. I don't know how to answer that. Probably.
Very philosophical.
It is very philosophical. I mean, watching things change in the ocean rapidly
Mhm.
makes me a little bit worried for the future, but I also wonder, so I actually think that 4Ms might be resilient to climate change. Has that made me like change how I do things? No, I don't know. I feel like I'm a pretty careful about things. Yeah, so I'm not sure. I think more in academia, I think what's changed is probably trying to change how we like take care of students and teach and mentor.
Mhm.
And now that I've been doing this in my position for 9 years, and then watching other researchers start their jobs, sort of that I think has been the thing that's probably changed the most for me.
And kind of in that vein, like someone asked, like, what is your favorite aspect of being a mentor, or do you have any advice for those who want to become mentors in the future?
Yeah, I guess that's whether or not you want to mentor students,
Yeah.
colleagues,
That's true.
right? I think it varies depending on what you end up wanting to do. And so I feel like you're doing a program where you're learning some teaching.
Yeah.
And I did not have that opportunity, and we have the same program at Oregon State. It's called the GCUT, which is the Graduate Certificate in Undergraduate Teaching. And you have something similar here. And I didn't have that. And so when I started teaching, I didn't have anybody teaching me how to teach. You just sort of get thrown into it in academia, where like if you become a teacher, you get a teaching degree and you learn pedagogy and you learn. And so we don't often get that in academia. And then you get a teacher and you're like, wow, this teacher's terrible. And that's because I don't know how to teach.
Yeah.
So I think we're trying to change that and make the options better and mentor our incoming faculty better, make sure that they aren't sort of thrown to the wolves and sort of to try to
Mhm.
sink or swim, because that's not fun for anybody.
True.
And then I think in terms of mentoring students, I just think that there's a lot we can do to help students in graduate school that we used to not do at all, which is,
Mhm.
having things like individual development plans where like
Mhm.
you actually plan your term. I didn't do that. Nobody taught me how to do that. I didn't even know these things existed until I had a student in another program. And she walked in and said, I need you to sign off on my IDP. And I said, what is an IDP? I don't know what an individual development plan is. Completely changed like how I teach my students and mentor. And I feel like that should be part of like all the stuff that we learn if we're going to become an academic, professor.
Mhm.
I don't know. I sort of went all over the place with that answer, but
These are just leading questions.
Yeah.
Want to talk
Yeah.
about. Yeah. And just a little plug for that program here is the ETE, the Empower Teaching Excellence or something. Very good program. So would recommend. Yeah. Questions? Cool. Do you have any personal or professional goals that you're hoping to attain, like going on into the future? Just kind of sticking with this paragraph.
Yeah, I mean, I'm not tenured yet. We have this really broken system in my college where when you become an associate professor, just in my discipline group, you don't get tenured until you're a full professor. So everybody else in my college, except my discipline group, gets tenure at associate. It's an old broken system, so that's kind of weird. So hoping to get tenure. I'm like an tenured. It's up or out, so I technically have job security, but I don't have the label of tenure. It's super soft. It's a little bit broken. I'd like to change that. There's a life goal, is to change that, to get rid of this archaic system that we have in my college. And I actually just started a business, which is this crazy side job that I've started. I owe you new slides, by the way, because we've improved our process. Yeah, so I should send you some new ones. But when we process forams, we put them in these little paper slides that they're called micropaleo slides, and nobody's making them in the United States anymore. And so I decided to make them. And so I'm trying to run a business.
It was a lifesaver for me because I was about to make them myself and I'm
Yeah.
not crafty enough to do that. So
I've heard that other people are trying to make them too, and everybody hates it. And we hated it at first also, but we have figured out how to make them
mhm,
and make a lot of them at once. And so things are working.
mhm.
But so we just recently made like, I don't know, something like 10 sales in this last month. So I feel like things are rolling. But I think that I would like for that to succeed.
Yeah.
Otherwise, I just have a very nice laser and and I will figure out other things to do with it.
It's always used for a laser somehow.
Yes, there's always a use for a laser, or I'll sell it. But it's been really fun. Our college has a program called the Accelerator,
Mhm.
where they teach you how to run a business. And they do it because it's beneficial for the college if you have IP, like intellectual property that belongs to the college.
Mhm.
They want a piece of your intellectual property. And they don't get a piece of mine because it turns out it's not patentable. What I'm trying to do is not patentable because anybody can do this. And so they don't get a piece, but I still got to do all the training. So I benefited from this like 9-month program. I got a little bit of money out of it, which was nice.
Mhm.
I got mentoring from folks who run businesses. I learned a lot. I learned how to do a sales pitch, which was crazy. And so I don't know, if you ever have an idea, you should, run with it because it's fun. Yeah.
I know you also do some like outreach things with like making 3D scans.
I
of-
do. Mhm.
Yeah. Like, do you market that as well, or is that?
No,
just
but
like-
I've been asked to.
Oh, okay.
So I made, I have something like 100 3D forum models. I recently took them off my website. I have like the saddest story to tell. So I have an outreach website
Mhm.
and you could freely download these 3D models. And there's like all these disclaimers all over them saying these are for educational purposes only. I own the copyright. You do not have permission to like take these and, you could make jewelry with them. This is not something that I want anybody else doing. Like I created these models.
Mhm.
And someone made art with them and then claimed that they were theirs. And then presented the art at a museum, and then the museum contacted me and asked if I would relinquish copyright. So it was like this really strange situation. And then I found the models, like he took some and just took 3 models and called it art and put his name on it and said they were 3D generated. I was like, you did not create, like this is not your creation. It's like no one's creation, right? It's just a 3D model of a shell. So I had to take the website down. And I'm going to put it behind basically put it behind like a login. So you have to get permission and then you have to say you're not going to do something like create art with it or ask me first. Like if this person would have asked me first, I probably would have been fine with it. But they wanted me to give them not only permission to have the copyright, but also to reproduce it and put it in books. And I was like, what? This is the craziest ask. But I have been asked to set up, like put 40 like plankton models together and sell them. And I have not done this yet because I'm too busy. But,
Yep.
At
Seems like
some
you've got
point
a lot of
I have enough going on. But people have asked me if I would do that.
How do you make those models?
A micro CT scanner. So we take actual micro CT like you would CT scan your body. And we have a micro CT scanner. We can add, forams from other periods of time. It's about $800 for a scan, and we can do about 80 forams at a time at a resolution of about 2 microns.
Wow.
So very high resolution, and then we process the data into 3D models. Yeah, and it depends on like there are different, so like the Bruckers have a, some of them have like a more 4 micron. So you might get the resolutions different depending on the model. But yeah, you can scan them here and make your own.
Yeah.
Yeah, or
Like, ooh,
so
love
the
that.
rads, if they're really thin, you need something that I think our instrument can go down to 0.8, and then the newer micro CT scanners can go down to like, 0.8, like 800 nanometer scale, and then you can get really fine scale stuff.
Good for me.
Yeah, they're really fun, and the little micro CT scanners have become like way more popular and cheaper. So I think our college had one of the first, and they built it from scratch for college because now you can just buy them.
Any questions about anything we've talked about so far?
Question, did
Oh.
you always want to be a professor, like when you went back to grad school, was that your goal? When did you decide that? What did you want to do with PhD?
Oh, you want to know something? I went back to grad school because I was working and I left science and I worked in the IT field. So a really weird trajectory in general. And I made websites. I did e-commerce and I had to wear a suit and like dress up and I liked it for like the first 2 years. I made a lot of money. And then I decided that I was working for like a cult. I don't know. Things just got really bizarre. I was telling someone at lunch today, I got in trouble for wearing a beige suit by like the only female like top boss because I wasn't dressed like a man. And so I should have been dressing the part. It was like the most offensive conversation. I didn't look like the top brass. I should have been wearing blue or black. And I was like, I need to get out of here. This is not for me. And so I went back because I had been away from science for so long and then the market crashed. So this was in like 2000, 2001, somewhere around there. I could not find a job for like $10 an hour going back into like science. And so I decided to go back to school. In between that thought process, I also had a baby. So I went back to school with a 4-month-old in tow. So that made life a little bit complicated. But no, I did not actually ever think that I would be a professor. I think that just happened. My advisor was like, I have a PhD project. And so I applied for it. And then I was in a PhD program. And then I was like, what do I do next? I become a professor. So I'm like the accidental professor. Yeah, it was not my intention. I'm a first-gen college student. So like nobody in my family has a PhD. I'm the oddball.
I might pivot a bit if that's okay with you.
Yeah, yeah.
We have a lot of research related questions.
Cool.
The first one of which is, what is your all-time favorite paper and why? You have one?
Oh my goodness, do I have a favorite paper? I do, and I'm going to be like, I actually absolutely love Jim Zakos's paper from, was it 2001?
Yeah.
And part of the reason that I like that paper so much, actually, there's probably two, David Lee and oh my goodness, there's probably a lot. So David Lee, I think it was a David Lee paper, wrote one of the first like long reconstructions of temperature using forams. And so that was one of the first papers that I read where the wiggles from the 4M temperature reconstruction matched ice cores. And so that I thought was amazing. But this paper by Jim Zakos that was published in, I think, 2001 or 2005, that sort of it's 4M data that is global, and it's a global temperature reconstruction of like basically bottom water temperatures from isotopes. And what I like about that paper is that you sort of look through what are the big things that happened in the Cenozoic and why were things different. So like the opening and closing of gateways, changes in the carbon cycle, changes in productivity. And it's just a really fun paper to talk to students about paleoclimate. And so I teach a paleoceanography class and we use that paper and sort of like walk through the Cenozoic and we talk about the things that have caused that. I don't know if I have any other favorite papers. I've never been asked that question. I probably have other ones. I think that one just comes to mind because I use it for teaching. And I just sort of like it a lot for that reason.
I'm impressed in you, the author. I oftentimes don't remember the author of the paper I read.
Right?
I'll need to remember this question for future reference. Yeah, there's some questions on more ecology topics if you want to talk about those.
Yeah.
One of them is, have we gotten definitive answers to any big questions in macroecology? Do you have an answer to that?
Oh, I don't think I can answer that question. Like macroecology in terms of what, like, I think that might be I think that might be not specific enough for me because I'm also not an ecologist.
We are adjacent.
We're adjacent, yes.
Our ecology center. Ooh, this is a fun question. Are there paradigms in ecology that you think are holding us back from progressing and making conceptual breakthroughs? Can we answer that one?
Oh,
Good
I don't know if I know how to answer that one either. I guess I think I would have to relate it to like the work that I do. I think that for a really, really long time, people have really just thought temperature sort of rules the roost for where organisms like forams live,
Mhm.
that it's just temperature. And this is going back to like, just looking at the community structure. I was actually just talking to someone else here about this, but it's not just temperature. So in our field, that's been like the main thing, what drives community structure in forams. It's the temperature tolerance that they live in. You know, pH plays a minor role, salinity plays a minor role. And the first climate reconstructions that were done were looking at assemblages, right? Like CliMap looked at what assemblages were there, what temperatures do we find them in, what temperatures do we find those, where do we find them in the past? And that must be what the temperature was like, and issues of not having like completely similar community structures. And I think the problem with that is that we're not taking into account the things that they interact with, right? We're not taking into account what do they eat, because we still don't understand what they eat. We're not taking into account the fact that things like productivity could change where we find certain species. And so I think that integrating, and I talk about my colleagues who are ecologists about this all the time. They study, I have a colleague that studies phytoplankton. And she studies phytoplankton using something called flow cytometry. So she runs samples through a flow cytobot and gets the distribution of what's there, and does lots of machine learning and neural network type stuff. And we can correlate then what is she finding versus what am I finding versus what are we finding in the sediment traps, and start to correlate the community structure that goes beyond just like what am I looking at, right? It's like insular to look at just the forams without considering the other stuff. And then correlating that piece to satellite data, I think is going to be the next boom in terms of like studying I guess just looking at planktic organisms. Because if we know what they're associated with in terms of plankton, we can use satellites that are expanding in their capabilities to look beyond just chlorophyll, but look at what organisms are creating, you know, different types of light interactions. I don't know, I think that we're just at the cusp of being able to do more. Yeah, I think that's it. I think that, and I mean, I'm thinking of it from a very like foram perspective, but community structure, I think, matters. They don't live in the ocean by themselves.
Very true. Yeah. A more specific to your field question is like, and I know you talked about this in your talk yesterday, but for maybe those who may not have been there, but like, why non-spinous foraminifera? Like, why are you interested in those organisms, and why are you interested in them, like, in general?
Yeah, that's a good question. So when I first started my postdoc with Howie, number one, he was like, literally, hey, come to Catalina. No one cultures the non-spinos. And I had been studying them for my PhD. And I had started making these image maps of their geochemistry using a microprobe. And there's been this sort of picture of how we think they grow calcite. And I remember showing this picture to my postdoc advisor and saying, the way that we think they grow, the way that papers have cited the way that we think they grow forever is wrong. And drawing the picture and saying, this is not what we see in the geochemistry. Why is this? And he says, because nobody else has done it. And so everybody's citing this paper from 1957 on how we think they grow from benthic forams. And so I think I lost my train of thought. I have to bring you back to the question. Oh, why the non-spinos? So I was looking at the way that they grow and thinking there's just stuff that we need to do to understand this better. And so that's where the culturing came from. And I grew them in isotope labels and grew them on day-night cycles to try to label calcite that grew. We came up with a different foram model. I think many of them are different. So my model doesn't, like, we wrote a paper on it, but my model doesn't answer how they grow for every one of them. And I just think that from the perspective of being able to understand where they all live, we have to know why are they biomineralizing different, and how can we use that better? And even in looking at how climate change is going to affect them, if we don't understand where they grow, how are we going to know where they end up? I don't know. There was just like this piece missing in the field. They weren't getting enough love. I think they just needed some more effort, and other people are looking at them. And then in the polar regions, several of the polar species are also non-spinos. There's, I think, only maybe one polar species that has spines. Yeah, so I mean, I was just, driven by like nerdy curiosity of trying to figure out a little bit more about them.
I was also wondering if you could describe to the room and for those on Zoom, like, what culturing is and what it entails. Like, what does it mean specifically for like plankton? Because I know like oftentimes people think of like petri dishes for culturing Like if you could expand on that.
Yeah, that's a good question. I'm actually about to give giving a talk in March on this, and I haven't thought about this in a while. I was just asked, like, why do we grow forams?
Mhm.
So when we collect them from the plankton, we put them in a petri dish and we pick individual shells that we want. And we put the individual shells in individual jars, which sounds like a lot of work. And the reason that we do this is, number one, if you put a bunch of them in a jar together, they tend to find each other.
Mhm.
Radiolarians, I just learned from you, do this as well. And when they find each other, one of them wins. So the other one dies. So
They
that's
eat each
bad.
other.
They kind of eat each other.
right there.
And you can see all this like rhizopodia between them, and like one of them wins. And then we also, when they grow, they don't reproduce in the lab. We have not been able to barely ever get them to reproduce in the lab. So we grow them from juveniles, and I want to only have the chemistry of the parts that grew.
Mhm.
And if I have 10 in a jar, I can't keep track of what got added. So we have to have them in their own little home. We have to feed them. We pay attention to what got added. We might add an isotope label. We might do things like transfer them in and out of different types of seawater. We can learn like how they're growing that way. Yeah, and then so for the different types, the one that I fed yesterday, for those of you who were in here, I pipetted like a little Artemia. We grow those in jars. The other ones go benthic. And I mentioned this in the talk, and that means that they sink and they settle on the bottom of these flasks. In order to find them, we have to put them in optically clear, like little Falcon jars. So we buy these flasks that they attach themselves to. We feed them in there, and then we wait until they die and bring them back. But we do that so that we can track every single specimen. And it's a pain, and it's tedious, but it's also kind of fun. You end up going somewhere that's, outside and the water and, collecting plankton. It's pretty fun.
I've always just been kind of curious why are those like Catalina Island and where I was in France and like on
Oh, yeah.
Taiwan, like where?
Why do we go where we go?
Yes, why do
That's a
people
really
go?
good question. So off the coast of Catalina, the really nice thing about Catalina Island and the reason that people go there, you are in blue water fast. Worms don't live you are not going to find high abundances in shallow water. And Catalina Island, the island is like this. It's like a tectonic part of California that's been separated. And so there's a little cliff that people dive even. We are in blue water in like 10 minutes.
Mhm.
So it's also really calm in the morning. We don't really go, like April's not fun. It's cold and the seas are rough.
Mhm.
So we go there in the summer, you wake up in the morning and the water's like glass. It's really, really nice. You get out into the ocean, you do your plankton towing and you come back and it's fast. Green Island is the same. And the reason that we would go to Green Island over Catalina is that it's different species, right? They're in the Kuroshio, we're off the coast of California, so you'd get different species. If you want to expand the diversity of what you study, you go somewhere else. Bermuda is a place where folks have been going for a while. It's a lot harder. I'm going to be there this summer. It is a 45-minute to an hour-long boat ride, and apparently everybody throws up. So I'm not looking forward to that. I'm going to be like, my collaborator can go. So I do not like being seasick. It takes me like a day or two to get better from being seasick. I do take Dramamine when I'm on ships. I don't take them on dive boats. I don't need it, but if I'm on a ship, I do.
Mhm.
And then, so for those reasons, and the same thing for you, like when you were in France, you get out into water fast.
Yes.
And that just makes life so much easier to get your collections done in the morning, get all your work done. Plankton tow days are long. You have to process samples, name them, feed them, measure them. I mean, sometimes we name them. But so that's a lot of work. And then off the coast of Oregon, we can't dive there and we can't use small boats. And so we have to be on ships. And that complicates things because seas are rough. And so, and that is just a complete luck if you're out on the water and it's nice. And if it's not, then you take a lot of Dramamine. Yeah, but mostly we like to go to these places because it's easy. Yeah.
I see some. Oh,
Where do you want to go?
One more time.
So we can collect the number that we collect in a day is a lot. We used to capture them. So divers would be tethered to the boat and they would capture individual forams and individual jars. And we don't do that anymore, partially because they wouldn't let us 2 years ago. They had a new dive safety officer and said nobody's diving. So we turned to plankton tows. And the reason that people would dive capture them is because they thought, well, they have lower mortality if you capture them in individual jars. But we've learned that they just need time to recover. So it takes 2 days. So people were giving it a day and saying, oh, these are not recovered, and dumping the water. They just needed a second day. So day 2, you get great recovery. So we will do plankton toes and pick them all into individual jars. And at this time, we do put a whole bunch of them in jars. Like we'll put 10 in a jar. They all float, go to the bottom, and then they regrow their spines. Or if I do falcon flasks, the ones that are spinos, we just put a bunch of them in, see which ones recover by putting out new spines. And we put them in what we call the ICU, which is just putting them in like a seawater table so that they can recover. So we might capture like, as many as we can feed the following day. So it might be like 100. That would be a good catch. And then we feed them every other day. And that's just based on habit. If we feed them every day, they die faster. Does something to like their cellular machinery. They sometimes don't grow as much. And then what was the other question? Okay, I think that was it, yeah.
Yep.
So we flew to Taipei, and I joked around that it was like a planes, trains, and automobiles type of trip. If you're familiar with that movie, we took a flight to Taipei. We stayed overnight in a hotel. We took a train to the southern part of Taiwan, and then we took a ferry to Green Island, which is where we did the work. And then on Green Island, the way that you get around Green Island is on a moped. And so we all drove mopeds for the entire like 5 weeks that we were there. It was so fun. Probably one of the most fun places I've been. It was extraordinarily challenging for eating. That was the hard part. But otherwise it was great. And so that's it. A plane, a train, a ferry, and a moped is how we got there. Mhm.
Oh, nice.
In the video, you're recording it during like 13-foot swell.
Yeah.
Like, looks in a microscope. And that's how awesome.
So there are some tricks when you're on a microscope and when you're on a ship, and one of them is to not use a lot of water. So we put, we put all of the material in like a crystallizing dish that's about this big, and then we swirl the material. All the forams go to the center. Take a couple of aliquots out into a petri dish. And the trick is to not add a lot of water, because if you add a lot of water every time the boat moves, everything goes back and forth. If you're seasick, number one, that's terrible. So we've learned that you just don't use a lot of water and kind of tilt it, and then the water can't go anywhere, and then you pick it like that. And then we take lots and lots and lots of breaks if there are high seas. And then sometimes we just, if it's really bad, we add some extra seawater to it and try to come back to it. And if you put them in, like add some extra seawater to it, sometimes we'll just take like the forams out of the bottom, all the hard stuff that settles, put them in a new Nalgene, add a bunch of fresh seawater, and stick them in the fridge. Like stick them in the walk-in, set the walk-in to like 16, and then go lay down and come back to it when things have calmed down.
Good question.
Yesterday, you mentioned how the community composition changes in relation to temperature, tropical species being seen in Oregon, et cetera. And I was wondering, do other factors that you've studied, like pH, also alter communities in this way? Or are the effects of those changes more related to individual shell composition?
That's a really good question. I don't know if we have enough data to figure that out. So pH makes them grow thinner, but it doesn't make it so that they can't grow there. I mean, I suppose if pH got really low, that would be problematic. But we've had pH events off the coast of Oregon as low as 7.4, like upwelled, really low pH water. We still capture forams in that water. So they're probably more robust than we think, but they all have a temperature tolerance range. And the interesting things that we've learned is that when they get outside of that temperature range, they become deformed. So that's fun. I should publish that paper. But yeah, I think it's mostly, I mean, I don't know if I answered your question. Right now, it's mostly temperature. I don't think I have enough data to look at stuff like productivity or things that they're living with to know what else is that factor. But I think it's mostly temperature. They have a salinity tolerance range. We do get low salinity. Most of our knowledge on that is people who grew them in culture and like lower the salinity to 17 or raise the salinity to 40 and figured out what do they do? At what point do they die? The algal symbionts actually probably have a temperature range. And so if it gets too warm, the forams bleach like a coral, and we can see the symbionts leaving the shell. So that probably plays a role as well. But I think it's mostly temperature. Yeah. They end up, so the events that we've captured this have been events that cause the temperature to decrease rapidly. And when that happens, they pop like aberrant looking chambers, small, thin. And then the one species, Orbulina, where we've seen the most morphological variability, they end up getting a morphology that is an extinct form, which is really fun, and with different apertures around the bottom of the shell. So they look like a species that went extinct in the Miocene. So completely different. It's like they can't form that last chamber, and they try, and then they form little apertures all around the base, which is really neat. So yeah, but we don't, and I think that people have done this in culture and have gotten them to also, they will pop funny looking, like I call them popping chambers, like making a chamber, becoming a little bit more deformed when they hit like their extreme sort of like happy range. Any follow-up questions? Yeah. Oh, I don't think so. There are ones that live in like briny water, so low salinity. The benthic ones can tolerate really low salinity. And I think, do you know if there are any? There are some that live in like, I think there is one freshwater species, but it probably lives in an estuary near saltwater. But they have tolerance ranges too, and so you can track sea level like transgression sequences by looking at how the species change through time. And people have done that like off the Oregon coast.
Really cool just hearing about this like intersection between like looking at modern stuff and like relating it back to a lot of these, because it's such a good fossil record of forams. Like,
Yeah.
just curious like how your work and your interests align between the modern and the paleo.
That's a fun question. So some of the things I think about in terms of like what we do with the modern, often you get these blips of like warm water species. Like there's a record off of the coast of the Santa Barbara Basin where like warm water species came in and they're very low abundances. Could that have been a marine heat wave? Right? And often those numbers will be less than 4%. You won't even include them in the assemblage. Will be part of the other.
Oh.
Right? And so if you look at that, it might change the way that we look at really brief excursions of warm water taxa, or brief excursions of cold water taxa, or excursions of like deformed species, which we see in the fossil record. That's an indicator that something changed rapidly. And I don't think I really thought about this until like Kelsey's work was done, and until we saw these really weird forams. That there's actually a paper from I think it's the Red Sea, where people found these extinct looking Orbulina, and then we saw them off the coast of Oregon in real time, and I was like, I bet you it was some weird event that caused that. So I would hope that it might change the way that people think about how we can interpret like these excursions that we typically ignore. I don't know. So I think that's been sort of something fun to think about. Yeah.
One other question from this sheet is, are there any ongoing or future studies you're particularly excited about that build on the work you're doing now, or just work in the field?
Yeah, so I have almost 4 proposals in review right now, which is a lot. But one of them is the SELP study. So going back and trying to look at these records and reconstructing SELP records and looking at calcite flux at the same time. So I think that'll be fun. That's a master's project. But the other one that I'm excited about is we're going to try to make marine snow. So that's a thing that you can do. You grow a bunch of diatoms. You get the diatoms to degrade on like a shaker table. We can grow the diatoms in like labeled seawater. So add a carbon isotope label and then track where that's going. So I think a question like you had asked, where is the carbon-13? Like what's happening to carbon isotopes? I want to know if what they eat ends up in the shell, what they eat ends up in the organic membranes. And I want to know if I can track the chemistry of like the marine snow particles. I want to make them and see what's happening. I want to label that process. And so that proposal is in. And then we also want to track, are they eating the marine snow particles? Because if they do, then they're playing a role in like a modification of the organic carbon cycle. They're actually eating some of the stuff that's being produced. So during periods of time where they're abundant, they might be actually changing their organic, like rain to the seafloor. That's all in the same study. And this was the first time that I've submitted a proposal to the biological oceanography program, and that one's got really low success rates for NSF, like 10%. So I'm a little bit worried that that one's probably not going to get funded, or it might get punted to chemical oceanography. But I'm kind of excited about that one. I want to make marine snow and track it and do some, I think we have plans to do correlative sort of TEM and nanosims.
Mhm.
So we can look at what's happening in the inside and track where the carbon's going, because the carbon will be labeled. So we can look at the sort of organelles on the inside and lipids and figure out where all of that stuff is going after they eat. I don't know. I'm really excited about that. And that's also very different from anything that, I do, right? Yeah.
Cool. Could I ask a bit about some of your secrets to success?
Oh, dear.
category?
I don't know if I have any.
You have some, definitely. How do you identify new areas of research? And feel free to take a drink of water.
By accident.
By
No,
accident.
no, honestly, a lot of it is having things not go right. Is like, I didn't talk about this. I had someone come up to me yesterday and ask me, like the jump from why did I decide to measure barium in the non-spinosporiums and do this partition coefficient experiment? And that is because I grew, the way that the folks grow spinosporiums is they add barium to label the calcite. And I did that. I was like, I'm going to grow forams and I'm going to add barium to label the calcite. Got back to the lab, analyzed the shells, and I knew these shells grew, but the portions that likely grow out in the open had way too much barium. And then I couldn't be certain that everything else grew in the label. Do you understand what I'm saying? Like the barium was what it should have been, but also the shell had high variable barium. And then I thought, I can't be 100% positive that these grew. Now I need to go back and figure out what they're doing with barium. I'm going to do this new experiment. I wrote a proposal, added an isotope label. And so that was just like having a bad field season and then being like, I have to figure out what happened and having it completely change my thought process on how they grow. So I think a lot of it's serendipity of having a discovery that is unexpected. And I feel like that's been half of my life, to be honest, is just being in the right place at the right time or having bad data and having to figure it out. And I tell my students this, that like failure is such a huge part of what we do, that I don't call it failure. Like I had a student come into my office. She was my first PhD student, and she came into my office and she said, my data are bad. And I said, why? And she was like, because it's not what I expected. And I was like, that does not mean your data is bad or your data are bad. I was like, it just means you don't know what's happened. And now we have to figure it out. And that turned into a cleaning study and a paper. So I don't know. I think that failure just happens. And I have a son who played baseball for like ever. And baseball is a game of failure, right? Like if you're batting 333, man, you're doing so good. 33% success rate at bat. So I say this because half of what we do in science is failure, right? We submit proposals that don't get funded. We submit papers that get rejected. We submit resubmitted papers that get rejected. I don't know. So I think part of that is just being tenacious and not taking it personally. It's a game of failure. Things are not going to go right. Just gotta okay, well, that stinks. Now we got to go figure out why.
That actually kind of covers that next question, which is about like obstacles and setbacks you've experienced during your career and like how you overcome them.
Yeah.
And is there anything you would have done differently looking back, and advice you would give to people that might encounter your position?
Don't take it personally. I think if I could have just had someone telling me that my whole life, like a little whisper in my ear, things are going to go wrong. People are going to feel not nice sometimes. And I don't know. My mom used to say, be like a duck in water. And I always hated that when I was a kid, like let it roll off your back. But it's kind of true, right? You can't take everything. And so I think I wish that somebody would have been like just whispering this stuff in my ear, because I took a lot personally. I think I mentioned I went to graduate school. I was 28 years old with a kid. And one of my first exams was a slash class with undergrads. And my first exam was just terrifying. And I got in the car and I cried the entire way home. So you just got to be like, sometimes things are hard and it's okay. Yeah.
Yeah. One of my favorite like internet memes is it's like, do you take constructive criticism? And then it's like, me already crying. Yeah, sure. Like, what's up? And it's like, feel that in my soul.
Yeah. Yeah, so I wish I would have just taken less personally, and I still do this where somebody says something to me and I go home and I ruminate about it for like days. I'm like, what did they mean? You know, it's like,
Mhm.
oh, stop ruminating.
Something we all have to learn.
Hmm.
Are there any unexpected lessons you've learned from your work? Like, I guess lessons being any topic of like research or just,
Oh my gosh,
Things
so
in
many
life.
lessons. I don't even know. I feel like it's all lessons. I don't know. I think it's just all of it. I don't know if I have any good advice on that. It's just that everything informs your next move to some extent. It's okay to do a reset. I had a 9-person lab group up until recently, and a whole bunch of my students graduated at the same time, and I'm taking a reset. And that's just what I told my dean. I have one student finishing in March. That's my last PhD student. I still have undergrads. I haven't I still have 4 undergrads working in my lab.
Mhm.
But I went from having 9 people, a postdoc, took on another advisor student, helped her graduate because their lab imploded. And I just need a break. I need a little bit of a lab reset, get caught up on things, and I think that's okay to do.
Yeah.
My other colleagues are like, good for you.
I just want to mention that we are getting closer to the end of time. We have 7 minutes left. So if anyone has burning questions or topics they want to cover, like, please feel free to mention.
Ask me.
I'm floundering up here.
You're doing great. You're not floundering.
At least outwardly, I guess not.
Yeah. So I've given public talks. I try to make them, I give a lot of background, right? Like, it's more of like why do we care about marine plankton, right? I think this is really, really important right now when we have, senators saying things like, someone got funded to study a marine plankton. I mean, that's not what they said, but they could, right? And then you have to be like, why is this research important? So I feel like this is critically important to talk to people right now. Go to your neighborhoods, we have a thing called tap talks, which is like talking at a bar. I've done those. Give a talk at your church, give a talk at your community center, give a talk to kids. So fun because they go home and they tell their parents all about forams or about whatever you're studying. I think it's so important for us to get into our community where we can make a difference. I've written to my senators no less than like 7 times in the last week. I don't think they're going to respond to me anymore, which is fine. But I don't think that that's going to help. I don't think that that's going to make a difference. I think if you want to continue to have education be important, we just have to talk to people. And so that's what I try to do. I mean, I've given talks at mostly things like tap talks, pubs. 500 Women Scientists have a series I've given talks at, but mostly it's trying to just get across the message of why what you do is important and how it relates to other aspects of our world, since sometimes it seems really obscure. Like, I study a plankton that's the size of a grain of sand. Who cares? And I did take some training. So we have a media training at the Oregon Museum of Science and Industry. And they talk about this message box. Have any of you heard of a message box? If you haven't, go and look up message box. The question that it's what are you, it has like this little box around it. I can't remember who created it. But the question that you keep asking yourself is, so what? It's like, I study marine plankton. So what? Like, it's important for climate. So what? Why? Right? You get down to like this nitty gritty. And that training was really, really helpful. And the message box tries to have you take your message down to something like simple. Not to make it simplified because people are smart, but to make it make sense to people who don't do what you do. That they're important for climate change. So they're gonna respond to climate change. They always have, and that they're sort of canaries in the coal mine, right? So they might be the first to go. We don't know that, right? Other researchers have looked at little marine calcifiers. I don't know how they're going to respond yet. If they go away, does it matter? It might, because we don't understand their community structure, but there are other organisms like off the coast of Oregon that people have studied called pteropods. And they're a little marine snail. They are eaten by salmon. And salmon is a huge industry off the coast of Oregon. Millions and millions of dollars. It sustains something like, this is in my student's current paper. I should know this. It's like 1.7 billion. I don't remember. Something significant supports the economy of Oregon. It's just the fisheries. You get rid of their food, we're gonna have a problem. You have disease strike the Dungeness crabs, we have a problem, right? So sort of getting that message across that it matters because this is part of the food web. And, coastal regions support 60% of the world's population. So trying to sort of, for those of you who like, study wolves and study cheatgrass and study, these are things that matter to people to understand. Hey, if these things go away, if they change how fires work, they do ultimately matter. And I think that's just that message that you have to get to people who don't do the research. The so what's hard. That's why. That's why it's fun to, have your elevator speech. What do you do? Have a 3-minute explanation of what you do. Something that you would tell someone in an elevator.
Do you have any advice on how to build that of yourself, or is it just kind of
Practice.
practice?
Practice it with each other. Practice it. They have little of like lightning events. Have a lightning event with your students, with all of you, where you come up here and give your 3-minute speech. Have a competition. Yeah, I think they do that at AGU. They have like a 3-minute elevator speech. They used to anyway, where you have to come up and get people excited about what you do in 3 minutes. Elevator ride. That's all you have is to get someone hooked. Yeah, I think it's just practice.
I'll make sure I let the audience have a chance to have a question before I ask probably what might be one or two more.
Okay.
Do you have like a topic that you want to discuss to end it out? Yes, I want to do that before I feel like, it's a random question.
I don't think so. I feel like you guys asked some good questions. Yeah. No.
Or just for those who are kind of interested in doing things like you, like, do you have any like advice for like ideal preparation and background for someone who wants to follow into like the same research path that you have?
Yeah, I mean, I think about what I tell my students and what I try to tell. I think it's really, when I went to grad school, the path was academia. That was the only path that I was told was possible. And that if I didn't make it, I was part of this leaky pipeline, right? This is what my trajectory was going to be, you'll be a professor. And when I was having a hard time getting a job, I remember just feeling like, feeling like a failure. I had gone through years of applying and not getting, getting interviews or not getting any interviews. And so I think diversifying your skill set so that you have multiple trajectories and none of them are failure, right? You can change your mind, right? I was always told that changing your mind was, even for graduate students, we have graduate students who opt not to complete their PhD or who take an extra year and they think, well, everybody else finished in 2 and somehow they don't measure. Don't compare yourself to anybody else. I don't know. I wish that I was just told that it's okay to change your mind, but also to build your skill set. And so I tell my students, like, if you learn the mass spec and you learn how to run the mass spec, you can go and do other things. Like you could work in the wine industry and run a mass spec, right? You can work in the drug industry and run a mass spec. It's a skill. So you might say, oh, I know how to analyze forams. No, you know how to run a mass spec. It's different. So it's like figuring out what that skill is rather than what you're analyzing, right? And that way you can branch out and you don't feel sort of like, hey, if I change my mind, it's failure. I don't know. People are allowed to change their mind.
That's great advice to end on. Can everyone join me in thanking you for
Yeah, thanks everybody. I feel like I haven't thought about these like philosophical questions. So I'm gonna go like sit like this for a little while.
Well, thank you guys all for coming and we'll be able to.
Yeah, thank you all. This was fun.